Hi, everyone. My name is Francois, and I'm a PhD candidate in the Integrated Remote Sensing Studio, which is part of the Faculty of Forestry at UBC. Today, we'll be discussing remote sensing and how it can be used in the tree improvement programs of British Columbia. First, I'll discuss where we see a space for remote sensing in the tree improvement cycle, followed by an introduction to remote sensing technology. We'll then look at a local case study, followed by our ongoing and future work. So as a quick review, tree improvement programs aim to change gene frequencies for a few key traits. For example, in Douglas fir, we're interested in yield. Within the tree improvement cycle, base populations are defined and phenotypically superior trees are selected. These trees need to be tested, with the goals being to evaluate genetic quality of the selection and to quantify genetic gains. While successful, several limitations to the testing exist, such as measurements that are labor-intensive, expensive, difficult, and time-consuming, and they only focus on taking very basic measurements. These measurements don't really tell us how trees are structurally or functionally different from one another. Remote sensing technology can address several of these limitations, as well as augment other parts of the tree breeding cycle. So broadly speaking, we can split remote sensing technologies into two categories, passive and active. Passive remote sensing technology detects naturally occurring energy. An example of this would be a regular camera. In contrast, active remote sensing technologies have sensors that emit energy, which is then reflected off a target and measured by the sensor. Airborne laser scanning, also known as LiDAR, is a great example of this. In the past, these technologies have been limited by their spatial scales, and as such, were suited to large area analyses. Satellite imagery was limited to 30-meter spatial resolution, for example, Landsat, while early ALS systems were limited to 2 to 5 points per square meter, which was good enough to characterize stands of forests, but not individual trees. Improvements in remote sensing technology have meant that we're now able to capture data in new and exciting ways, such as with unmanned aerial vehicles, or UAVs. This means that we can collect data more frequently, while technological improvements mean that the data can be captured at higher resolutions. For example, UAV imagery can be captured at low single-digit centimeters, while LiDAR data can now be collected at hundreds to thousands of points per square meter. This is an example of an ALS system we used in 2018 on the right, which was mounted to a UAV. This improved technology has given scientists the opportunity and ability to phenotype individual trees. We can not only provide proxies for traditional measurements, but also derive new metrics that describe structural and functional differences between trees in improvement trials. For example, in the figure on the right, we can calculate tree height and diameter, but we can also calculate things like volume based on airborne laser scanning. So, what is ALS? ALS is an active remote sensing technology where a unit sends pulses of light towards an object, which is then reflected back. This can be seen in the GIF on the right, where the red points are reflected back to the sensor. This allows us to produce a very accurate three-dimensional point cloud, as we know exactly where the sensor is in time and space. ALS produces tree heights and is also able to characterize the ground very well. There are two different approaches that we can take with airborne laser scanning, the area-based approach and the individual tree crown approach. The ABA has historically been favored as we had low point densities, and that was the best way that we could characterize vertical structure in the forest. However, as technology has improved, we can now turn our attention towards individual tree crown approaches as well. Digital aerial photogrammetry is a passive remote sensing technology and utilizes concepts from photogrammetry. Pixels are located in overlapping images, which we can plot in 3D space to form a point cloud. DAP provides structural and spectral information, and we are able to use a variety of sensors, such as normal cameras, which provide RGB imagery, all the way to multispectral and hyperspectral sensors, which provide further information from different portions of the electromagnetic spectrum. In addition, DAP is generally very cheap to acquire. Here we can see an example of a DAP point cloud with some pixels that have been matched from successive images. The workflow from going from a point cloud to an individual tree is very similar for ALS and DAP. Processing these data sets requires an understanding of the data set itself and the forest that you're working with, as the parameters will vary. This is an example of a point cloud that we got in 2018, and it's been clipped, classified, normalized, cleaned, and finally segmented. It's at this stage that we can produce metrics for each tree, ranging from tree height to more complex metrics. So now that we know the technology, we can talk about how we've used it in our research in the context of a case study, namely realized gains trials of coastal Douglas fir. We gathered data for 5 sites and tested both individual tree and area-based approaches. Our aim was to look at variation in crown structure between trees of different genetic levels, as well as comparing our findings with field measured data. Here we can see our study sites, which are spread out in coastal BC. The pink crosses are the realized gains trials, and they were planted in 1996. This is an example of what one of the field sites looks like. You can see the different genetic levels planted throughout the site. Using an individual tree approach, we created a suite of metrics aimed at finding differences between the genetic levels. Here we can see a Weibull probability density function that we created, and you can see that there's variation between the top cross trees in the green and the wild sand trees in the red. Our findings indicated that genetically superior trees were generally taller with higher, shorter, and denser crowns, but that interaction effects between genetic level and spacing were very important. We also looked at detecting individual branches in the upper third of crowns, creating metrics such as branch volume, angle, and width, as you can see in this workflow. We found that the top cross trees were consistently different, but again, spacing caused interaction effects to take place. Taking a different approach, we looked at the realized gain sites using an ABA to see how well we could predict ground measured gain. As we can see from the figure, our gain predictions on the x-axis were generally very good when compared to the field measured gain. This model uses metrics representing realized gains for leaf area index and mean canopy height to predict volume. Currently, our lab is working on the Douglas fir trials, in particular, looking at how we can use remote sensing in the progeny trials. And we're also working with interior spruce to look at changes in crown form and foliage spectral reflectance as predictors of growth in trials across BC using DAP. Here we can see a map of some of the field sites that are scattered across the province, and you can see an example of a field site here. The imagery for these sites include regular RGB imagery, as well as multispectral imagery and LiDAR, as we can see from left to right on the slide. Here we can also see what they look like in cross-section. Finally, the outlook for integrating remote sensing data into tree improvement programs looks extremely bright. Data is becoming more accessible in that it's becoming cheaper and easier to collect, while processing is getting simpler all the time. Companies are creating tools that improve the workflow from raw data to the finished product, while free and open forestry-specific packages are becoming available. An example of this that we use is the LiDAR package, which is available in R. Finally, I think it's important to note that we're now at a point where we have enough experience with different technologies that we can focus on the strength of each technology. And by picking the right tool for the job, we can maximize the information that we can derive from a single acquisition. We have published some of this work already, and we're looking forward to publishing more as we go along. And we'd just like to say thank you to you for listening, and also to our funding partners who've made all of this possible.
Hello, everyone. I hope all of you are safe and healthy. So it's my pleasure today to share part of my research from the CEDAR project. So Dr. John Russell, the founder of the BC breeding program for Western red cedar, he established this project with collaboration with the Balman Lab at UBC, and the main objective is to deliver elite selection to industry seed orchards decade ahead of traditional breeding by applying genomic selection. So he started the program in the late 90s by wild stand selection of 1,000 parent tree, and he was working on improving growth, hardwood durability, and deer browsing resistance. So it has been proven that some secondary metabolite, mainly terpenoid compound, are responsible for this durability and browsing resistance. And as you can see, the most difficult trait to phenotype is the hardwood extractive because we have to wait for 25 years, while we are able to get an accurate measure for 4-year extractive for 1-year-old seedling. So in general, breeders are interested in getting an accurate phenotypic measurement from a progeny trial to get an estimate for the breeding value, rank the individual, and select the best one for deployment and seed production for reforestation. So the main driving force of our project was to predict the hardwood durability at the seedling phase. So this is a coastal program now. Dr. Marie Vance started a new program for the interior. So the basic idea of genomic selection is to predict the phenotype from the genomic data at any age from a prediction model. So in order to do that, we are collecting the genomic data and phenotypic data from an old progeny trial. And then after fitting the model, we are testing this model. This is what we call a model validation to get an estimate for a prediction accuracy, which measures the predictive power of our model. After that, we can simply plug in the genomic data from our target population in the model to predict the breeding value and then make selection. So the basic idea is that we are doing early selection by using information from an old progeny trial. So in our project, we selected our training population from a first generation polycross progeny trial, and we selected 15-20 trees representing 26 polycross family, which means that we know the mom of each individual, but not the dad. But the dad is coming from one of these 21 males used in the polycross. And we phenotype this population to the 3 traits. And our objective is to predict the phenotype for our target seedling population, which is another first generation offspring, but it is a full seps. And it's sharing some parent with our training population. And we phenotype this population for the foliar extractive because again, we are able to get an accurate measure for this trait at this young age. So first, we tested our model in the training population for all the traits, and then we tested our model in the target population for only foliar extractive, again, because it is the only trait that we are able to measure at this young age. First, we use our molecular data to do a pedigree reconstruction and paternity assignment to identify the dad of each individual in our training population, which reveal a significant unequal male contribution. And you can see that we have a lot of variation in the number of offspring per individual male. And we found that we have 5% pollen contamination. So this pedigree reconstruction now converted the polycross design into the full SIPs. Now we have more than 400 full SIP family, but they are small and unbalanced because again, we have a lot of variation in the full SIP family size. Second, we use the genomic data in the analysis to get an estimate for the breeding value. This is what we call genomic blob to overcome the limitation of polycross. So polycross design is mainly designed for a backward selection from the female parent only, and it is not accurate for forward selection. But using the genomic analysis to get an estimate for the breeding value, now we get 2 advantages. So the first one, we increase the selection intensity for the backward selection because now we have also the male breeding value. And then we can use it for forward selection because we get up to 22% increase in the breeding value accuracy for our offspring. And also it resulted in up to 35% increase in the expected genetics gain. For our prediction model validation in the training population, we validated the model in different scenarios. So the first one is the most flexible. We call it random tenfold cross validation. And here we randomly divide the data into 90% to fit the model and 10% to test the model. This is what we call the validation population. And we got a really high prediction accuracy for all the traits. So the prediction accuracy is measured on a correlation scale. And then we validated the model in a more restricted scenario across the three sites, the two generation, parent and offspring, and different ages. And we got a small decrease in prediction accuracy. For testing our model in our target population for this analysis, I used the alpha-tizone, the major terpenoid compound in the foliar. And the meaning of this analysis that we are using the training population to fit the model, but we are testing this model in our target population. So this prediction accuracy here, it comes from the validation in training. And within our target population, we have two groups of seedlings. So the first group, which is around 40% of the population, they are sharing both parents. So they are highly related to our training population. This is why we got a small decrease in prediction accuracy. While the second group is sharing only one parent. This is why we have more decrease in prediction accuracy. So here I am sharing with you the breeding value for wood extracted for the best full sip predicted family. And within this family, we have 15 seedlings. So we are also able to use a pedigree for making prediction. But in this case, we got only one value, which we call it the family mean for the 15 seedlings. But by using genomic selection, we are able to have a predicted individual breeding value for the 15 seedling. And you can see here that we have a lot of variation around the family mean. And this is actually what genomics is enable us to do in the absence of phenotype. So now we are able to do selection within this family. So after the model validation, we apply the model to predict the growth rate and wood trait for this target population. After that, we constructed a multi-trait selection index for the two predicted traits and the one measured trait. And then we run an optimal selection to balance between the genetic diversity and the genetic gain. And we selected the best 100 individuals to construct the seed orchard. And also Dr. Albin Chuck decided to move the whole target population for field testing to validate genomic selection in the field. And actually, this is one of the first application of genomic selection here in Canada. As a final conclusion, we have two main advantages for molecular breeding. So the first one, in the presence of a measured phenotype, using molecular breeding or genomic analysis is giving us a more accurate estimate for the breeding value. So in our study, we overcome the limitation of the polycross mating design, and now we are able to use it for forward selection. So the second advantage is to predict the phenotype at the seedling phase and for multi-trait early selection. So in our project, this advantage, coupled with the fact that seedling younger than 1 year can be reproductively induced in Western red cedar, these two factors together are reducing the breeding cycle into only 2 years, which is translated to increase in genetic gain because now we are saving the time required for progeny trial. So finally, I would like to thank the Bowman Lab and the BC Ministry of Forest, all collaborators and co-authors, and for the conference committee for giving me this valuable chance. And I am more than happy now to answer your questions in real life, and don't forget to stay safe and healthy. Thank you.
Hello everybody, my name is Jean-Philippe Laverdière. I'm a master's student at Université Laval with Jean Bousquet and Patrick Lenz. This presentation is about the integration of drought response into multi-threat improvement scenarios for white spruce. The climate conditions in the boreal forest are dramatically changing. The increase in annual temperatures coupled with constrained period of precipitations will augment the intensity and frequency of drought events. The latter is mainly related to the weakening of the jet stream, causing large oscillations of the polar vortex into lower latitudes during winter and Gulf of Mexico heat waves during summer at higher latitudes with more frequent weather blockages. Several studies have underlined the negative effect of drought episodes on the radial and apical growth of boreal tree species. They are also known to increase the susceptibility of trees to pests and disease by weakening them. Intense drought can even cause mortality in young plantations. Our study focused on the impact of drought events on radial growth. However, extreme climatic conditions that will be faced would not be constrained to heat waves. Extreme cold temperatures, more frequent thaw period, and spring and early fall frosts are other examples of climate extremes that may occur more frequently. White spruce is among the boreal tree species on which the most frequent and intense drought episodes will have a negative effect. The species' habitat covers a considerable proportion of the boreal forest, ranging from Alaska to Newfoundland and Labrador. It is important to the Canadian pulp and lumber production industry due to its superior mechanical properties. The tree breeding programs across the country have mainly focused on growth and survival threats at the beginning. In the subsequent cycle, adaptive and resistance threats as well as wood quality have been considered sporadically. Just recently, the resilience to climatic stresses, such as drought, is gaining interest for integration into breeding programs. But in order to do so, the genetics of resistance and resilience to drought needs to be investigated. To analyze drought impacts on standing trees from common garden experiments, retrospective tree ring analysis have recently been used. As you can see from the wood disc on the left, tree rings width varies from year to year and reflects optimal or suboptimal growing conditions, mostly due to climate. The lower graph on the right represents the evolution of annual basal area increment of different families in a recent publication. The authors have associated 3 growth reductions with reduced summer soil water availability as represented in the upper graph. We use the same methodology to detect drought episodes and then inspect the genetic control of tree response to drought. For this study, we used a 19-year-old white spruce genetic trial that is replicated in 3 contrasting environments in the province of Quebec: Normandin, Valcartier, and Watfeld. The inspection of climatic variables related to the monthly humidity having an effect on radial growth revealed 2 different drought period stresses. These periods were in 2010 at the site Normandin and one in 2012 at the site Watfeld. Unluckily, no such periods were observed at Valcartier, and a thinning of the Watfeld test was also conducted in the fall of 2012. The effects of the drought stresses on the radial growth are presented in the 2 figures. The upper graph presents the annual radial increment. The lower graph presents the detrended radial growth for both sites. The growth reductions of 2010 at Normandin and 2012 at Watford were matching higher drought indices than normal conditions. For this study, we estimated 4 drought response threats according to these authors. The resistance, resilience, and recovery threats are illustrated in the figure for 2 different individuals. The first, resistance, refers to the radial growth loss observed due to the drought event. The second, recovery, refers to the radial growth upturn observed following the drought event. The third, resilience, refers to the capacity to reach the same radial growth performances after the drought event as those observed before the drought event. Finally, relative resilience is the resilience relative to the growth loss observed at the time of drought. We could only inspect the threat resistance at the site Watford because we could not consider the period after the drought due to the thinning of the test that was conducted after the growing season affected by the drought event, which interfered with the normal recovery reaction of trees after the drought. Besides the 4 drought response threats, we also analyzed diameter at breast height, tree height, wood average density, and conducted acoustic velocity measurements, which is a good indicator for wood stiffness. The accessibility to dense genomic information allows us to consider the use of genomic prediction to estimate genetic parameters as well as assessing genetic gains. Instead of using pedigree information, the use of genomic profiles can lead to less biased estimates of heritability and genetic gains, reduce the duration of breeding cycle, and facilitate multi-threat selection. A genomic selection approach needs in the first step a number of phenotyped and genotyped trees to build predictive models. After model training, cross-validation is performed to evaluate model accuracy. Once the models are validated, they can be used on non-phenotyped trees to predict breeding values based on genomic information only. Using both conventional and genomic selection, we estimated heritability and analyzed correlations between the threats. Finally, we estimated the expected genetic gains resulting from multi-thread selection scenarios. This figure shows the narrow-sense heritability estimates obtained at both sites for the threats that we assessed. As you can see, we only calculated the resistance threat regarding drought response at Watford due to the thinning intervention that may have influenced growth recovery in the following years. At both sites, we obtained somewhat lower heritability estimates for the drought response threats than for the growth and wood propriety threats. The genetic control was therefore low to moderate for drought response threats as opposed to moderate for the conventional threats. To evaluate the efficiency of both pedigree-based and genomic models, we calculated their predictive accuracy of breeding values. At both sites, both methods show lower accuracies for the drought response threats compared to growth and wood threats, with the exception of tree height at the site Normandin, which was low. Also, we found that the accuracy values for the growth and wood threats were lower than those found for multi-site analysis in an earlier study. This is because of the reduced number of trees used in this single site analysis here. Finally, the model accuracy was similar for the conventional and the genomic selection approaches. We also analyzed the correlations between drought response threats and growth and wood threats. Surprisingly, the two sites presented opposite correlation regarding the threat's resistance and DBH. For example, at the site Normandin, the correlation was negative. This indicates that the bigger trees at Normandin were less resistant to drought. On the other hand, the correlation was positive at the site Watford, meaning that the bigger trees were more resistant to drought. There were several differences between the sites that could explain these opposite patterns. For example, the sites did not suffer the drought episodes at the same age. Also, the drought intensity and moment in the growing season differed at both sites. The sites were also abandoned farm fields which might have differed in their drainage. In order to consider drought response in addition to wood quality and growth threats, we developed various multi-thread selection scenarios. The objective was to improve all threats, but the priority was the height genetic gain. We found that the integration of wood density and drought resistance both at 20% along with height at 60% of weight could fill this need. The scenario permitted an improvement of all threats at both sites, even if we found opposite correlation between some threats. At the site Normandin, the scenario permitted a 12% genetic gain compared to the maximum of 13% obtained for height-only selection. At Watford, we obtained a 9% genetic gain for height compared to the 10% maximum gain. This slight sacrifice permitted a positive gain for all threats. An important genetic gain in DBH was also obtained for both sites as it was highly correlated to tree height. In conclusion, we obtained a low to moderate genetic control for drought response threats, with resistance showing lower heritability than recovery, resilience, and relative resilience. The accuracy of genomic selection models for drought response were slightly lower than for wood proprieties and growth threats models. Even if opposite correlations were found between drought resistance and DBH at the two sites, a selection scenario considering small weights for density and drought resistance permitted to obtain a positive genetic gain for all threats on both sites. This work presents an important step toward the integration of drought response to white spruce breeding programs, which appears as a necessity in the context of climate change. Finally, because drought episodes may vary in time and in space, and because tree response can also vary with age, site-by-site and drought-by-drought analysis is warranted for the time being until we know more about genotype by environment interactions affecting drought resilience threats. I would like to thank Patrick Lenz, co-director of my master project, Simon Nadeau, Martin Perron, Claire Depardieu, and Jean Bousquet, my research director, that all have participated with enthusiasm to the realization of this study. This project was supported by the funding from Genome Quebec, Genome Canada, and the Canadian Wood Fiber Centre. I also thank the Canadian Forest Genetics Association for letting me present here. I will be available to answer your questions later. Thank you.
Hello everyone, my name is Iman Rashidijouybari, a PhD candidate at Laval University. Today I'm going to present part of my study titled as Multi-Trait Selection for Improved Solid Wood Physico-Mechanical Properties in White Spruce. In this study, we focused on white spruce, which is extensively planted in Canada, nearly 170,000 hectares per year. Its natural distribution spans the North American continent from the Atlantic to Alaska. In fact, the species is among the most important boreal conifers. Its wood is highly valued by the Canadian forest industry due to its physical properties such as low density but high bending stiffness and strength. Still, there are some drawbacks. As a boreal conifer, it has a long rotation age, despite its importance for industrial applications and studies on negative correlations between growth and wood quality. Management policies and breeding programs rarely considered wood quality. Several applications are introduced by spruces, such as building constructions, desirable source of fibers for papermaking industries, or interesting chemical components for bioproducts. However, its main application remains as construction and building materials because of its wood properties. In the province of Quebec, structured breeding programs for white spruce has started in 1960s. Primarily, they have focused on improving growth rates. In some cases, adaptive traits such as cold hardiness, insect and disease resistance have been considered, but only limited studies have focused on wood quality traits. In general, conifer breeding programs were focusing on growth and increasing the productivity. They also decreased rotation age at some point. But breeding can also help to produce trees with more valuable and cylindrical stems, or even select for wood properties and reduce their variability in order to augment wood quality. There are a few genetic studies on wood quality in spruce with promising results for their inclusion into tree breeding programs. And only a very limited number of genetic studies actually considered flexural wood traits, which are the main traits for structural applications. With this motivation in mind, we obtained a dataset of direct measurements on solid wood samples to address the following objectives and to better understand the genetic of flexural wood traits, for example, their genetic control and the possibility to obtain genetic gain. Then, to understand the genetic relationship between wood flexural properties as well as other wood quality and growth rate. And finally, evaluate the effect of selection among traits. We recovered logs from 300 trees of a thinning intervention in a polycross trial. The samples are from two different sites located in contrasting environments in the province of Quebec. Each site was subdivided into 4 blocks where trees were arranged in single tree plots. We analyzed trees from 38 families with reconstructed pedigree. As you can find in this slide, we spotted the middle part of the stem just above breast height for flexural wood measurements. The statistical model used in this study is animal model. To obtain flexural properties, we first obtained planks from each log around breast height. They were stored in a conditioning chamber for 2 months until the moisture content of the samples stabilized. Then 2 samples from each side, north and south side, close to the bark, were prepared. Finally, samples were stored in conditioning chamber right before testing. Overall, we prepared close to 600 samples, so preparing 600 samples and testing was quite time-consuming and intensive work, but we did it. The measurements used in this study were flexural properties known as MR and MOE, wood density, and ring width obtained by X-ray densitometry, height, acoustic velocity, and diameter at breast height. In the above pictures, you see the samples are actually broken in a testing machine, and the force and deflection is measured. This imitates the case of structural applications, where we are interested in the modulus of elasticity as MOE and modulus of rupture as MOR to evaluate the load resistance, while MOE is a measure of the stiffness where elastic deformation is reversible, and MOR is related to maximum strength that a piece of wood can resist. At first step, we obtain the actual mean values for each trade at different trades. In this table, I only present a subset of trades. In summary, we noticed that wood quality trades were higher in site Valcartier, while higher radial growth were obtained at Normandin. This is mainly because of the fact that the spacing at the site Normandin was slightly wider than in Valcartier. In this slide, genetic control of wood quality traits as well as genetic gain are presented. Based on the results, genetic control of wood quality traits are stronger than tree growth rate. Wood density and MOE were found to be under moderate genetic control, 0.46 and 0.38, while MOR is under low to moderate genetic control. Type B correlations were high for all traits, and we didn't observe any G by E interaction for wood quality traits. The genetic gain varies between 6.9 for the density and 16.5 for bending stiffness. The gain estimates are encouraging for selection of wood traits. Similar gains were obtained for MOR and height. It should be noted that the smaller genetic gains estimated for tree growth trait is due to a lower heritability, whereas heritability is higher for most other traits. In this slide, Table 3 presents among trait genetic and phenotypic correlation. MOE and MOR were strongly and positively correlated to wood density, while only low to moderate phenotypic correlations were found. It was to mention that the correlation between flexural properties and tree height and DBH were low to moderate and negative, which is inconsistent with other studies. Here we see the correlated response between traits. For example, if we would select for height due to correlated response, we would obtain -28 and 33% of genetic gain that could be obtained by direct selection on MOE and MOR. Selection for density, on the other hand, will result in efficiency close to 100%, meaning that virtually the entire genetic gain of MOE and MOR could be realized through indirect selection based on wood density. According to our results, selection based on height and especially DBH lead to increased flexural properties. As a conclusion, flexural wood properties are genetically controlled and considerable genetic gain could be expected when considered in selection. And a positive, moderate to high correlation exists between flexural wood properties and density. This means that opening up an opportunities to indirect selection of wood quality. And moderate and negative correlations between flexural properties and DBH and height suggest that selection for stiffness and strength may lead to lower volume production. And finally, combining multiple traits in a selection index will help to obtain the best compromise for all traits of interest that corresponds to breeders' priorities. And finally, thank you for your attention and have a great day.